Good evening, Gautam. Good evening. Good evening. Okay, let's wait a minute. Let others also join fast. Good evening, Deepika. Good evening. How are you doing? Good evening, Savita. Okay, friends, just listen. We have completed up to unit 4, right? Good evening, Ananya. We completed up to unit 4, right? Unit 1, 2, 3, 4. We did not do the unit 5 also. We did not do the unit 6 also. And today actually we have to do the unit 7. Today I will try my best to take both unit 6 also, unit 7 also. Okay, we will try it. If I couldn't take, what I'll do is tomorrow I will try to take undoubtedly unit 7 as well as the unit 8. Today itself I will try my best to take both units, unit 6 and unit 7. If I couldn't complete, then we will go for the unit 7 and 8 tomorrow without a fail. Okay, so I appreciate your patience today, because a lot of questions to be solved. Okay, I'm good, Deepika, thank you. Okay, good evening all, good evening all. Let's continue, let's start. Okay, please type start. We will go ahead. Please type start. Okay, let's go for the question number 1. Okay. Yes, friends, your time starts now. 30 seconds. Your time starts now. 10 seconds more. Stop. Stop, 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 stop. Okay, give the answers fast, first. The answer for the question number 1 is the C. What's the question says that gibberellic acid controls the seed germination by direct breaking down of the stored starch. In which one of the following tissues of the barley seed alpha amylase gene is induced in response to the GA? Suppose example, this is a starchy endosperm. So the starchy endosperm having the outer layers. What do you call this particular outer layer, friends? We call this particular outer layer as aileron layer. We call it as what? Aileron layer. Very good. The question number 1, the answer will be C. Get ready for the question number 2. Ready for question number 2. Ready for the question number 2. Very carefully you have to think and answer these kind of questions. Your time starts now. 1 minute. Question number 2. "50 seconds more." It's a very logical question, friends, but it's easy question. 40 seconds more. 20 seconds more. 10 seconds more. Stop, 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 stop. Friends, anyone, come on, give the answer. Question number 2. Okay, no one, no one. Question number 2, what is the answer? Question number 2. Okay. Question number 2B. Anyone else? Anyone else? Okay, I think almost all people are wrong, friends. The answer for the question number 2 will be A. Look into the given question. PCD, programmed cell death, also appears to occur during the differentiation of the xylem trachea elements that leads to the nuclei and chromatin degradation. These changes results from the activation of certain genes. Following are certain genes encoding. The question they says that which one of the following combination of the above is involved in the differentiation of the xylem trachea elements. Friends, during the process of differentiation, majorly we require the topoisomerase, because friends, this particular chromatin is going to get degraded. I mean to say that, friends, chromatin undergo great coiling. So to remove that particular coiling from the chromatin, I think one fellow has given the correct answers, Akash. What happens? Chromatin undergo the coiling, a very deeper coiling, I mean to say. To remove that particular deeper coiling or to remove the tension, we will use the topoisomerase enzyme. Option A is absolutely right. And see here, it leads to the nuclei degradation also. Name itself indicating, my friends, nuclei is getting degraded. Then what kind of enzymes are involved in the degradation of the nucleus or nuclei? You know that we require a certain kind of proteasomal complexes also. But in case of a nuclear degradation, the major enzymes involved is the nuclease. And you will get a doubt, sir, why you did not choose the answer protease? Because friends, in case of a nuclei, protease cannot be directly not involved. You know, na? See, there is a difference. It is not a proteasome. Friends, please kindly don't get confused. It is not a proteasome. It is a protease. Proteases are the one basically cut the peptide bond, so it cannot be involved. The enzyme which are involved in the nuclear degradation is what, friends? Obviously nuclease only. Hence the correct answer will be A as well as the B. Then the answer for the question number 2 is the option A. Simple question. Okay, friends, get ready for the question number 3. No problem, no problem. Remember the answer next time. Get ready for the question number 3. Your time starts now. 1 minute. Friends, think and answer. It's a very big question but easy question. Time starts now. Thirty seconds more. Twenty seconds more. Very simple, friends. 15 seconds more. 5 seconds. Stop, 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 stop. Okay. The answer for the question number 3, which is a straightforward question, will be the D. They are asking a question, friends. You can see here the membrane of the thylakoid divide the surrounding chloroplast into two spaces. we call thylakoid space. I have told you many a time. What do you call thylakoid space? Means lumen. And this stroma, they are the site of light dependent reactions of photosynthesis. The question asked is, which of the following processes is most affected? When thylakoid is damaged, the interior of the thylakoid is no longer separated from the stroma. Especially friends, thylakoid means what? You know, right? Thylakoid membrane is the region. Imagine this is the thylakoid membrane. On the thylakoid membrane, what are present? Different type of electron acceptors are present. And most, see, you know, right? Photosystem 2 is present. Photosystem 1 is present, and several electron acceptors are present. Most importantly, F0 F1 complex is also present. F0 or F1 complex, we can also call it as what, friends? ATP synthase enzyme, which is involved in the synthesis of ATP. So that can be majorly damaged. Hence the correct answer will be what, friends? D. Perfect. Get ready for the question number 4. Don't be hurry, slowly. Question number 4. Your time starts now. 1 minute. It's a very simple question. Think and answer. "40 seconds more." Stop. Okay, anyone has given the answer? Good. Question number 4, the answer will be C. Okay, what's the answer for the question number 4? The answer will be C. See friends, you all know very well, in case of a symbiotic nitrogen fixation, integration of the symplasmid of rhizobium into the root nuclear genome. First of all, friends, there will be no integration of the symplasmid occur. That's not even possible. The next one, sensing of plant flavonoids by rhizobia. We read it very clearly. What we called this particular flavonoid? So we will be calling them as a chemoattractants. Then option C, activation of NOD genes in the rhizobia. Obviously true. Activation of NOD genes in the legume roots. We have discussed very clearly. There are two type of genes are there. Sometimes, friends, NOD genes we can also call it as what, friends? NOD factors. Correct, na? NOD genes are NOD factors present in the bacteria, and NOD genes present in the leguminaceae plants. But both are involved in what? Nitrogen fixation only. They have asked the combination of correct. A will be wrong. Obviously B is right, C is right, D is right. Hence the correct answer will be C. Simple question. Friends, get ready for the question number 5. Question number 5. You can look into the explanation very clearly. Question number 5. Your time starts now. "20 seconds more." Okay, Savita has given answer. Gautam has given answer. Okay, Okay, 5 seconds more. Ananya. Okay, great. Stop. Friends, the answer for the question number 5. The answer for the question number 5 is the D. That is A and C. It's a very direct question, right? Option A. Red light photoreceptors are represented by a gene family. Absolutely right now. So you know, right? There is a phytochrome A, phytochrome B, phytochrome C, phytochrome D, and phytochrome E. There are 5 gene families are there. We read it. Option B, phytochrome C is the most prominent photoreceptor to perceive light. Obviously wrong, right? Photochrome A and B said to be the principal or the major one, not at all A. Obviously wrong statement. Option C, cryptochrome 1 and cryptochrome 2 have evolved from bacterial DNA photolysis. I told you very clearly. Here DNA photolysis act as what? Chromophore and apoprotein. Here photolysis is the one actually acting as a apoprotein. Right? Option C is also right. Option D, far red light is perceived by phytochrome D. Not at all. Phytochrome A and B. Hence the correct statements are option A, option C. Hence the answer for the question number 5 is the option D, which is a straightforward question. Plant physiology mostly will get a very direct questions, but little questions are from the experimental based. Very good. Get ready for the question number 6. Let me know, please look into the explanations and kindly let me know if you have any kind of a doubts. Question number 6. Yes, your time starts now. Question number 6. Forty seconds more. Gautam says answer Saima. 10 seconds more. Stop. Okay, the answer for the question number 6, friends, it's B. Okay, now look into the question. Both reducing and non-reducing sugars are transported efficiently through phloem. No, right? Reducing sugars cannot transported through phloem. Only non-reducing sugars can transport through the phloem. Simple point. Option B, it is a wrong statement. Sucrose uploading can be both symplastic and apoplastic. I hope you remember. Loading also symplastic and apoplastic. Even friends, unloading also apoplastic and symplastic. Kindly do not forget this particular statement. Hence the option B is correct. The route of phloem uploading is mesophyll cell, where the sugar synthesized in the mesophyll cell, from mesophyll cell to parenchyma, parenchyma to companion cells to sieve tube. You know, right? Companion cell can act as a linker cells which connect the mesophyll cells to the sieve tube cell. B, C is right. Transport in sieve tube as per the pressure flow mechanism proposed by a bulk, we call bulk flow hypothesis or the Munch hypothesis, based on the name of the discoverer Ernest Munch. Then C, D also correct. Hence the correct answer will be B, C, D. The correct option will be B. Perfect. Get ready for the question number 7. Get ready for the question number 7. 1 second, 1 second, friends. Just 1 second. Just give me 1 moment. Yeah. Question number 7. Your time starts now. Time starts now. $40 more. $40 more. Fast, fast. Okay, 10 seconds more. Stop, stop, stop. No one has given the answer so far. Question number 7. Time up, time up. I don't know how. I don't know why most of the people... Option D is the correct. Okay, everyone look into the question. They are asking us very clearly about the scheme of electron transport. If you quickly recall P680, after the P680, what is the immediate electron acceptor? Pheophytin. From the pheophytin to plastoquinone A, plastoquinone B, and immediately what? Cytochrome B6F complex. So they have given PC. So option A is a wrong statement. You look into the option B, P700, immediately what we can call it as a phylloquinone, iron sulfur A, iron sulfur B, iron sulfur X, FD, FNR, finally NADP plus. Obviously, option B is the right one. Now look into the P680, pheophytin, quinone A, quinone B, cytochrome B6F, plastocyanin 700. Obviously, C is also right. Then P700, phylloquinone, iron sulfur XAB. No, right? SA, SB, SX. Right. Option D will be the wrong option. Then what will be the correct answer? No. Something I said wrong. Huh? C is D, right? Iron sulfur XAB. I'm so sorry. Correct. Right. Option C is correct. Pheophytin, quinone A, quinone B, cytochrome B6F, PC700. Right. P700, phylloquinone, which we also call it as A1. Then iron sulfur X, A, B, F, D, F, N, R. Right. Option C is correct. Option D is also correct. Hence the correct answer for the question number 7 is C. Right. Question number 7 is C. Right, correct. Okay, get ready for the question number 8. Okay, your time starts now. 1 minute. Okay, I'll read this if anyone is unable to see here. Tracheids, vessel elements, sieve tube, sieve cell. Every single statement we have studied in the classroom. Option 1 says that cheap water conducting elements in gymnosperm. Vessel elements, cheap water conducting elements in angiosperm. Sieve tube, food conducting elements in gymnosperm. Sieve cells, food conducting elements in angiosperm. It's a very direct and straightforward question. 1 minute. $30 seconds more. $20 seconds more. Okay. Stop. Okay, the answer for the question number 8, it will be A. It's a very direct question. As we read that whichever is highly developed, they are always present in the angiosperms, because angiosperms are the most advanced plants which are completely and highly developed in the nature. So you look into the question carefully. They have given tracheids. You know, right? Tracheids are what, friends? They are not highly developed. So tracheids are most commonly present in the gymnosperm. And you know, right, vessel, the name vessel itself suggesting what? Angio. So the angios vessel elements are exclusively present in the, where it is present in the angiosperms. Now if you look into the sieve tube, tubes are highly developed. Whichever highly developed present in the angiosperms, sieve cells which are low developed, they are present in the gymnosperm. Hence the correct answer will be A. You people always remember, whichever is a highly developed, they will always present in the angiosperm. Whichever is low developed, they are always present in the gymnosperm. So please kindly don't confuse with that. Which highly developed angiosperm, which little developed in the gymnosperm. The correct answer for the question number 8 is option A. Okay, get ready for the question number 9. Okay, your time starts now. 1 minute. Think and answer. It's a very simple question. 30 seconds more. Okay. 10 seconds more. Stop. Please give the answer. Question number 9. D. Okay. What about others? Fast. Friends, it's a very direct question, right? Not even one. D. Okay. Next. I'll give one more second time. The answer for the question number 9, it will be B and C. I don't know how people has given option A as the correct. Everyone look into the question carefully. Option A says that pentose phosphate formed by the photosynthesis. You only tell me, why does pentose phosphate formed by the photosynthesis? Photosynthesis always give rise to what? Triose phosphate. Why will it give rise to the pentose phosphate? It never ever give rise to the pentose. Obviously statement A itself wrong. Now you look into the option B. Carbon stored as a starch, which is a polysaccharide, exists in the chloroplast, at night, and in the form of maltose, and it is converted into sucrose in the cytosol. Because we read very clearly, the site of production of sucrose is where? Obviously in the cytosol only. Where is the site of production of starch? We read it is occur in the chloroplast stroma. Obviously from the chloroplast stroma it is going to enter into the cytosol, where it is converted into sucrose. Sucrose is the one which travels through the phloem. Option B is absolutely right statement. Now you look into the option C. During short distance transport, sucrose move from producing cells in the mesophyll to in the vicinity of the sieve elements in the smallest veins of the leaf. Even in the classroom, we read the same statement. Even I reminded you. These are all the statements directly given from the standard reference book Lincoln and Tace. Because why we use the word short distance transport? Once the sugars transported through the phloem, they just has to enter into the sink region. That's why we can call short distance transport. Obviously C is right. If you look into the option D, in the process of phloem loading, sugars are transported into phloem parenchymal cell. No, right? How does the sugars transported? Sugars transported via sieve tube cells. Then parenchyma cell is absolutely wrong statement. Hence B is correct. C is correct. So the correct answer will be undoubtedly B. Very simple. Answer will be B for the question number 9. Friends, please slowly think and answer. Don't be hurry. Get ready for the question number 10. Questions are very big but very logical, very simple. Fast. Which one of the 30 seconds? That's all. Question number 10. 20 seconds more. Ten seconds more. It's an easy question. Stop, 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 stop. Anyone? Okay. Akshay, look into the question. Which one of answer for this particular question number 10 is the option A? Which one of the following compound is not a part of the alkaloid class of secondary metabolites? We discussed very clearly what are the various kinds of alkaloids. See, sorry. You know, right? Nitrogen containing compounds are of four types. Nitrogen containing compounds are of four types. What are those? One we called as alkaloid. Correct na? Second one we can call glucosinolate. Third one we called cyanogenic glycoside. Then fourth one, non-protein amino acids. Correct na? Now if you look into the lignin basically comes under what category? Lignin comes under phenols. It's not a nitrogen containing compound, or it is not an alkaloid. Whereas indole, tropane, pyrrolidine, these are all giving rise to what? They are all giving rise to atropine, they are giving rise to codeine, they are giving rise to morphine. These are all what? Alkaloid, but this is a phenol. Hence the correct answer will be A. Okay, friends, get ready for the question number 11. Your time starts now. 45 seconds, not much time for this. Your time starts now. Question number 11. Friends, 15 seconds more. Ten seconds more. Stop, stop, stop. I hope at least one will give the answer. Okay, look into the question. Which one of the following best describe the symplastic? I don't know what you are answering. They have given very clearly, right? Symplastic. How many number of times I have told you? The moment you heard the name of symplastic, only one point has to come in your mind. Symplastic means plasmodesmata. Why you are thinking about that? What is plasmodesmata? Plasmodesmata is the one which connect the cytoplasm of the two cells. That's what we can call the plasmodesmata, is the cytoplasmic bridge. So suppose example, all of you listen, look into the diagram very carefully. Suppose this is the cortex. These are all what cells? Cortical cells. Correct na? These are all the cortical cells. And this is what? Endodermis. Endodermis contains what? Casparian strip. Then finally this will be the xylem. How the water can move? By using the symplastic pathway. See here, plasmodesmata is present. Correct na? So through the plasmodesmata, water molecule enter. But if it is an apoplastic, I have told you very clearly in the classroom, through the cell wall intercellular space. Through the cell wall intercellular space. That is what apoplastic pathway. Symplastic means inside the cell. So which is what do we call symplastic. The correct answer, and direct one. You no need to look into other options also actually. Water travel across the root cortex via the plasmodesmata. Very simple, friends. Direct question. Question number 11, the answer will be B. Slowly, slowly. Get ready for the question number 12. Get ready for question number 12. Your time starts now. 45 seconds. We discussed this. It's a direct question. Please kindly don't make it wrong. Thirty seconds more. Friends, 15 seconds more. 10 seconds more. Stop, 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 stop. The answer for the question number 12 is the B. The herbicide dichlorophene dimethylurea. Sometimes instead of giving dichloro-diphenyl-methylurea, they'll give other name. What is the other name for this? You can also call it as a diuron. Correct, na? So what is the ultimate function of dichlorophenyl dimethylurea or diuron? It can directly block the electron transport between two photosystems. That is between P682D, P700. It's a direct and straightforward question. The answer will be B. Simple. Get ready for the question number 13. Your time starts now. 1 minute. Friends, don't get confused. They have asked a question which is incorrect. 30 seconds more. Okay, stop, 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 stop. The answer for the question number 13 is option A. Friends, you all people can easily remember the answer. DCMU always block the electron flow photosystem 2. First point. Then second point, you have to remember paraquat always block 1, photosystem 1. So if you remember this, you can answer the question easily. Option A says that both DCMU and paraquat block the electron flow in photosystem 2. No, right? Only DCMU. Option A is wrong. Both DCMU and paraquat block the electron in photosystem 1. No. DCMU blocks electron flow in photosystem 1. Absolutely wrong statement. The only correct statement is what? DCMU blocks the electron flow in photosystem 2, and while paraquat blocks in photosystem 1. This is the correct statement, option D. But what the question they asked is identify the incorrect statement. So option A, that is A, B, C. Very, very simple question. Very good. Correct, brother. Get ready for the question number 14. Your time starts now. 30 seconds. Friends, 15 seconds more. Stop, 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 stop. The answer for the question number 14, one second, is option A. Option A. Because MYC2 can act as a positive gene regulators. Positive gene regulators. Whereas JA, sometimes you can call JAJ. Okay, anything is same. JAJ, jasmonate gene domains. They are basically act like a friends negative gene regulators. They are acting like what friends? A negative gene regulator. Option A will be the correct. Very good, very good. Okay, get ready for the question number 15. Your time starts now. 20 seconds for this question, that's all. Ten seconds more. 10 seconds more. Stop, 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 stop. Question number 15. Okay, the answer for the question number 15 will be the option C. Option C will be the answer. Parthenogenesis. Friends, gibberellic acid is the major one actually involved in this stem elongation. Parthenocarpy also, because if one is doing the parthenocarpy, how can it do the parthenogenesis? It is doing parthenocarpy seedless without fertilization or seedless fruits. It is not at all involved in the parthenogenesis. Malt production. Sometimes instead of asking the malt production, they will say that whether the gibberellic acid is involved in the brewing industry or not. See friends, even gibberellic acid plays a key role in the production of alcohol also, but not at the higher rate, at very lower rate. So it is present in the, it is involved in the brewing industry also. That is only we can call the malt. So it is not involved in the parthenogenesis. Answer will be C. Perfect. Okay, get ready for the question number 16. Your time starts now. 1 minute. Friends, they have asked which is not correct. Okay, don't be hurry. Oh, 1 second. 1 second. 1 second. 1 second. 1 second. Please start. Please start. They are asking which is not correct. "15 seconds more. Question number 16. Stop, 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 stop. The answer for the question number 16 is the B. Friends, they have asked which is not correct. I have already told you where the Casparian strip is present. Imagine this is the epidermis. After the epidermis contains cortex. What does the cortex contain? Cortical cells. And after cortical cell there is a endodermis. On the endodermis what is there? Casparian strip is present. Correct na? So they are majorly present in the endodermal region. They are asking which is not correct. They said that it is formed in the growing part of the root. No, it never grow on the growing part of the root. Majorly Casparian strip is present where friends? In the endodermis. Very very simple, you know, right? Endodermis is rich of lignin. Simple. The answer for the question number 16 is the B. Okay, get ready for the question number 17. Your time starts now. 1 minute. Very logical question, friends. Very important question it is. 30 seconds more. Please try to give the correct answer. Ten seconds more. Stop. Anyone, friends, quickly? Question number 17. Who can give? You have to tell me in 5 seconds, otherwise I'm going to tell the answer. It's a part C question. Okay, the answer for the question number 17 will be the D. Okay, they have asked about correct statements. I'm requesting all of you look into the question. No, everyone look into the question. The question says that glucosinolates are synthesized by the elongation in the length side of precursor amino acid. If you quickly recall my words, nitrogen containing compounds contains what? Alkaloid, glucosinolate, cyanogenic glycoside. They are all basically derived from what? All are exclusively derived from the amino acid. In the PPT we read a specific line. What is the specific line we read? All the nitrogen containing compounds are derived from the common amino acid. That is the statement we studied. Obviously option A is right. Option B, all terpenes produced in the herbs and spices are sticky, oily liquids which reduce the edible value. No. Why they reduce the edible value? Terpenes are acting like a feeding deterrent. They are acting against the herbivores and insects. Because friends, we read the statement very clearly. Secondary metabolites do not interrupt the growth, developmental processes of the plant body. Hence, it cannot decrease the edible value. Option B is wrong. Option C, cyanogenic glycosides are produced and stored in a toxic form in plants. If they are stored in a toxic form in the plant will die. I don't know whether you remember or not the same statements we discussed. Cyanogenic glycosides in the plant, they are intact. What do we call intact? Inactive form. Upon crushing of the plant or upon damaging of the plant only, they are going to be converted from toxic form, non-toxic form to the toxic form. Don't forget that particular statement. Hence option C is wrong. Option D, the defense molecule alkaloid or their precursor are gathered from plants and used by some insects for their own protection against the predator. That kind of species, what do we read? We called adaptive species. Correct, na? Which are completely dependent on them. So they can eat them easily and they can use that particular secondary metabolites for their defense because they are adapted. They cannot get affected by this particular secondary metabolites even though they are toxic in nature because they are adapted. Right? Option B is also correct. Don't get confused. It's very important. Hence the correct answer will be D. A and D are the correct answers. Everyone understand, I hope. Good. Next, get ready for the question number 18. Yes, your time starts now. 1 minute. Your time starts now. 50 seconds more. Friends, 10 seconds more. Please think and answer. Your time starts. Stop. Okay. Stop. The answer for the question number 18 is the C. Okay. Look into the condition. The transcription factor DREB1 and DREB2 bind to the cis acting. No, right? They are always trans acting. That's the only word is wrong. Remaining entire statement is right. They are completely transacting. Option A is wrong. ABA induces many genes such as LEA, late embryogenesis, abundant protein, and RD29. They are going to be synthesized only during the stress conditions. Otherwise, they don't release at all. Then option C, ABA responsive gene contains 6 nucleotides. Of course, true. Called as abscisic acid response element, whereas 9 nucleotides are present on the ORE. Correct na? So ORE is 9 nucleotides, there is no doubt, and ABRE is the 6 nucleotides. Of course true. Hence the B, C, D are right. Option only A is wrong. Hence the correct answer will be C. Simple, direct question. Correct. Okay, get ready for the question number 19. Your time starts now. Forty seconds more. 30 seconds more. 10 seconds more. Stop. The answer for the question number 19 is the B. Friends, answer will be B. What's the question they asked? Rhizobial genes that participate in the legume nodule formation are called as NOD genes. The NOD D encoded protein. I hope all of you quickly recall. There are two type of genes are there. NOD A, B, C. We call common NOD genes. And NOD E, F, G, H, Q, L. There are other genes are there. We can call it as a host specified NOD gene, common NOD gene. But we discussed very clearly, if all these genes wants to be active, first of all, friends, they all has to associated with what? They all has to be associated with a NOD D gene, because NOD D gene is the constitutive gene. Every time it is expressed, first it get expressed, then it will come and associated with a region we call NOD box. What is NOD box? It's a promoter. It's a promoter. NOD D will come and associated with this promoter. They induce the transcription of all other genes, that is common and host specified. Hence the correct answer will be B. Okay. Friends, is that clear everyone? See friends, in the PPT there are 4 more questions are left. I am requesting, we will upload in the Telegram. I am requesting all of you solve that particular 4 questions also without fail. In the tomorrow class, please keep it in your mind, we're gonna cover unit 7 as well as the unit 8. All of you listen, tomorrow there are total 20 questions will be given, 10 questions from the genetics as well as 10 questions from the human physiology. Okay. 10 questions from the human physiology, 10 questions from the genetic in the tomorrow class. Okay. And as I told you, I am going to do a class on the prediction of the question paper. That is on Thursday sharp 5:30 p.m. Without fail, I will do that. Okay. So please remember that. I will see you in the tomorrow 7:30 p.m. class. Thank you all. Thank you. I hope the discussion is little helpful to all of you. Thank you all. I'll see you in the tomorrow class. Thank you so much. Good night all. Good night. Thank you.